Welcome again to BioBits and Bytes.
Great to be back.
Today we're taking another deep dive, this time into the incredibly versatile world of CRISPR technology.
The big one. Lots going on.
You've given us a fascinating stack of information, everything from, the nuts and bolts at Adgene to cutting-edge research on RNA imaging and the intricacies of CRISPR-Kingism systems.
It's a lot to cover for sure. The field moves so fast.
So our mission today is to synthesize all of this for you, highlighting the most innovative ways CRISPR is being applied beyond just, simple gene editing.
Right. Think of it as CRISPR 2.0 or maybe even 3.0 at this point.
Think of it as your shortcut to understanding the next wave of biological manipulation and discovery. We'll be covering everything from more precise gene surgery and controlling CRISPR's power to large-scale genome rewrites, RNA targeting, and even watching RNA molecules in action inside living cells.
Yeah, that live cell imaging stuff is particularly exciting.
Prepare for some real insights.
Okay, let's get straight into it. CRISPR has moved way beyond the original Cas9, right? And a major area of expansion is the development of new Cas9 variants and orthologs.
Orthologs. So basically different versions nature came up with in various bacteria.
Exactly. You can think of them like that. Cas9 enzymes from different species.
Okay. And the Adgene materials detail several Cas9 enzymes with more flexible PAM sequences. Now, the PAM, that's the protospacer adjacent motif. It's that short sequence Cas9 needs to see to know where to cut. So what's the real advantage of having Cas9s that are less picky about this PAM?
What's fascinating here is that relaxing that PAM requirement, it dramatically broadens the regions of the genome we can actually target precisely. Think of the genome as this vast library, and the PAM sequence is the specific address label a standard Cas9 can read.
Right.
Enzymes like SCas9NG, CPCas9NG, SPG, SPRY, and SPRYC. They can read more addresses. Essentially, gives us access to previously inaccessible parts of the genetic code.
So more doors unlocked in that library.
Exactly. PRY, for instance, can recognize NRN or NYN TAMs, which just opens up a significantly larger editing landscape.
And that's critical when you need a very specific change, but the original Cas9's preferred PAM just isn't anywhere near it.
Precisely. It's like having a master key that fits more locks. It's really about increasing the scope and precision of where we can edit.
That's a great analogy. More flexible keys to the genome. And then there are the Cas9 orthologs you mentioned, like SACS9 from Staphylococcus aureus. Adgene points out it's smaller.
Yeah, that's a key point.
Making it great for delivery, using adeno-associated viruses or AAVs. Why is that size difference so important there?
Well, if we think about gene therapy, AAVs are basically the delivery trucks we use to get therapeutic genes or gene editing tools into cells.
Okay.
But there's often a size limit, a packaging limit, to what you can cram into these viral trucks.
Right, the payload capacity.
Exactly. SACS9's smaller size means we can more easily fit it into AAVs. It overcomes a potential bottleneck in actually delivering these powerful editing tools where they need to go for, say, therapeutic purposes.
Makes sense. And Adgene also mentioned HSP1-Cas9 and NMEP2-Cas9. Higher fidelity.
Right. Similar size to SACS9, roughly, but they offer higher fidelity than the original SACS9. And higher fidelity basically just means they're less likely to make mistakes.
Fewer off-target cuts, for sure.
Exactly. Less likely to snip the DNA somewhere you didn't intend, which obviously increases the safety profile for gene editing.
And FCas9 with its unique NNTA PAM sounds particularly useful for plant genome engineering, opening up new possibilities there.
Yeah, expanding the toolkit for different organisms too.
So, okay, we have more versatile cutters, smaller delivery-friendly ones, and more precise ones. What's the next level of control in this CRISPR toolbox? How do you sort of manage all this power?
That raises a really important point. How do you put the brakes on these incredibly powerful gene editors?
Yeah.
And that's where anti-CRISPR proteins, or ACRE proteins, come into play. As Adgene explains, these are naturally occurring inhibitors of CRISPR systems.
Inhibitors, so like nature's own off switch for CRISPR.
Kinda, yeah. Think of them as nature's way of controlling gene editing systems found in bacteria.
So these ACRE proteins are like safety switches. Where do they actually come from?
They've evolved in phages. Those are viruses that infect bacteria and other mobile genetic elements. These things often need to evade the bacterial CRISPR defense systems to survive,
An evolutionary arms race.
Pretty much. And what's truly remarkable is the variety of ways these ACRE proteins work. Yeah. Some physically block the guide RNA, the cure RNA, from loading onto the Cas9 enzyme so it can't even find its target.
Okay.
Others directly inhibit Cas9's cutting activity, its endonuclease domain, even when it's already bound to the target DNA.
Wow.
And some makers even go as far as to basically flag the Cas9 protein itself for destruction, triggering its degradation.
That level of control sounds incredibly valuable. Why would we want to actively stop CRISPR once it started doing its job?
Well, the main reason is really to improve safety and precision.
Okay.
If Cas9 remains active in the cell for too long, it increases the chance of undesirable off-target, edits those changes in the DNA at locations other than the one you intended.
Right, the unintended consequences.
Exactly. Or it can even just be toxic to the cells if it hangs around cutting for too long. By using ACAR proteins, we can potentially time it, precisely dictate when and where Cas9 is active, limiting these potential side effects.
So it's like having an emergency stop button or maybe even a timer for your gene editing machinery. Finer control.
Much finer control, yeah.
Okay, so we can now target more genes with better Cas9s and then control their activity with ACAR proteins. But what about making more intricate changes to the genome than just cutting a gene or maybe, disabling it?
Right, that's where things get even more sophisticated.
Yeah.
Add gene details homology-directed repair, or HDR. This allows us to introduce very specific genomic edits.
Like changing a single letter in the genetic code or inserting a new word.
Exactly. HDR allows the cell's own repair mechanisms to do this using a DNA template that we provide. This template contains the desired new sequence, and it's flanked by homology arms sequences that match the DNA on either side of the target cut.
And those arms act like guides.
Precisely. They guide the repair machinery, telling it to incorporate the new genetic information accurately at that specific spot.
And the length of those homology arms is crucial, right? I think Adgene mentioned that.
Absolutely. As Adgene points out, if you want to insert a larger piece of DNA, you generally need longer homology arms to make sure the repair process is efficient and accurate.
But HDR isn't always the best for really big insertions, is it?
No, it can become quite inefficient for very large pieces of DNA. And that's where these CAST systems come, and CRISPR-associated transposon systems.
Okay, Cas systems. What's different about them?
What's fascinating here and kind of counterintuitive is that the Cas effectors in these systems, both type I and type VK, they don't actually have a nucleus activity. They can't cut the DNA themselves.
So how do they work?
Instead, they team up with transposase components. These are proteins like TNSA, TNSB, TNSC, TNAQ, to insert large donor DNA sequences. We're talking potentially exceeding 10 kilobases, which is pretty big.
Wow.
And CRISPR guides this whole transposon machinery to the target location. It's like CRISPR acting as a highly specific GPS to guide a mobile DNA insertion vehicle right to the spot you want in the genome.
So CRISPR guides, transpose, and inserts, a big leap for making substantial genomic modifications.
Huge leap, yeah.
And then we also have CRISPR recombinases and integrases mentioned, another way to do large edits.
Yes, these are another class of enzymes that can perform large-scale genomic edits, usually by inserting DNA at specific landing sites within the genome.
Landing sites.
Predefined spots. And the really ingenious part is how CRISPR can be used with these. There's a technique called passage that's prime editing assisted site-specific integrase gene editing.
Okay, that's a mouthful, passage.
It is. But what it does is use prime editing, which is another very precise CRISPR-based technique to first install these landing sites exactly where you want them in the genome.
So you create the dock first.
Exactly. Then an integrase enzyme like EvoBXB1 or EBXB1 can come along and efficiently insert the larger DNA sequence into that specific predefined site. It offers an incredibly targeted way to introduce substantial amounts of new genetic material.
So we've gone from making maybe single-letter changes with HDR to inserting potentially huge chunks of genetic code with castids, and now precisely targeting large insertions using these integrase systems after creating a landing site. The genome editing capabilities are becoming incredibly refined.
They really are. The level of precision and scale is increasing dramatically.
Okay, let's shift gears a bit. Let's focus on using CRISPR for a different purpose: screening. Adgene mentions high-throughput CRISPR screening with libraries. What exactly are these libraries?
Right. So think of these libraries as massive collections of guide RNAs.
Yeah.
Each gRNA in the library is designed to target a different gene in the genome. Maybe even all the genes in the genome.
A whole toolkit targeting everything.
Pretty much. Adgene provides these libraries either as pools of liquid DNA or sometimes as ready-to-use lentiviruses. The fundamental idea is to introduce this huge collection of gRNAs into a population of cells.
Okay.
And then see what happens when thousands of different genes are individually knocked out or modified across that cell population. It's a way to systematically probe the function of tons of genes all at once.
So It's a powerful way to figure out what different genes actually do, like on a massive scale.
Yeah, exactly. It's discovery science accelerated. AdGene outlines the basic process. If you get a DNA library, first you need to amplify it, make many, many copies. Then you use next-generation sequencing, NGS, just to check that the library is good, that it contains the expected distribution of gRNAs.
Quality control.
Right. Next, you package this DNA library into lentiviruses. These viruses can then infect your experimental cells, delivering the gRNAs inside.
Okay.
Now, depending on your setup, you might need a separate way to get the Cas9 enzyme in there, maybe co-infect with another virus carrying Cas9, or often you use special cell lines that already stably express Cas9.
Got it. So cells have Cas9, then they get a specific gRNA from the library.
Exactly. Then you apply your experimental condition. Maybe you're looking for genes that make cells resistant to a particular drug, for example.
A selective pressure.
Right. After applying that pressure, you collect the cells that survived or showed the phenotype you're interested in, and you use NGS again. By comparing the abundance of different gRNAs before and after the selection, you can see which ones became enriched.
So if a gRNA targeting gene X is way more common in the surviving cells, then gene X was probably important for sensitivity to the drug.
Precisely. The gRNAs that are enriched point you straight to the hit genes, the ones whose modification caused the observed effect.
Yeah.
It's incredibly efficient for discovery.
Okay, really powerful stuff. Now let's talk about dCas9. I remember this is a version of Cas9 that's been deactivated. It can't cut DNA, right?
No.
But it still has important uses.
Absolutely. D for dead, Cas9, catalytically dead. It can't cut, but the key thing is it can still bind very specifically to DNA sequences, guided by its gRNA.
So it's like a programmable DNA binding protein.
Exactly.
Yeah.
And because of that, it can be repurposed by attaching other functional proteins to it. This lets us do a whole range of things without actually altering the DNA sequence itself. AdGene highlights several of these applications.
Like what?
Well, for example, CRISPR and CRISPR. The I is for interference or inhibition. A is for activation. These involve fusing dCas9 to proteins that act as transcriptional repressors or activators. By guiding these dCas9 fusions to the promoter regions of genes, those are the control switches that turn genes on or off.
Right.
We can effectively silence genes with CRISPR or turn them on, activate them with CRISPR.
So instead of editing the DNA sequence itself, you're just controlling how that sequence is read or transcribed.
Right.
That's a fundamentally different way to manipulate gene function.
Precisely. It's about modulating gene expression levels rather than changing the code.
Very cool. What else can dCas9 do? Ajin mentioned visualization.
Yeah, this is really neat. By attaching a fluorescent protein, like the common green fluorescent protein, GFP, to dCas9, we can literally light up specific locations in the genome inside living cells. You guide the dCas9 GFP to your sequence of interest, and boom, you can see it under a microscope.
So you can track where bits of chromosomes are or how they move.
Exactly. It allows researchers to track the movement and behavior of specific genomic regions in real time within the context of a living cell nucleus. Seeing is believing, right?
That's like having a molecular flashlight that you can program to shine on specific parts of our DNA.
Amazing.
And it can also be used to purify specific regions of the genome. How does that work?
That's correct. Techniques like entry-engineered DNA-binding molecule-mediated chromatin immunoprecipitation or capture utilize tagged dCas9. Sometimes even the guide RNA itself is tagged.
Tag it how?
Usually with something like a biotin tag or a flag tag, small molecules that we have antibodies or other binding partners for. So you express the tag dCas9 and the specific gRNA in cells.
Okay.
The dCas9 binds to the target DNA locus. You gently break open the cells, and then you use the tag like a handle to fish out the dCas9 complex along with the piece of DNA it's bound to.
Like molecular fishing.
Exactly. It's followed by affinity purification. And once you've isolated that specific region of the genome, you can analyze everything that came along for the ride, proteins bound nearby, associated RNAs, even other DNA sequences that might be interacting in 3D space.
What kind of analysis?
Powerful stuff. Mass spectrometry to identify proteins, RNA-seq for the RNAs, and NGS for interacting DNA regions. AdGene emphasizes that these CRISPR-based purification methods often offer advantages in terms of simplicity and maybe specificity compared to older, more traditional approaches.
So dCas9 has really evolved into this remarkably versatile platform. It's not just a broken cutter, it's a whole new toolbox.
It really is. A Swiss Army knife for interacting with the genome without necessarily cutting it.
Okay, let's move on to another really exciting application area. Using CRISPR for diagnostics, we have information about RNA targeting with Cas13 and its use in pathogen detection. How does this work? Is this different from Cas9 targeting DNA?
Yes. Very different. Cas13, as Adjane explains, is a type dis CRISPR enzyme, and its key difference from Cas9 is that it specifically targets single-stranded RNA molecules, not DNA.
RNA.
Okay. It even has the ability to process its own precursor guide RNAs, which is kind of neat. But what's particularly interesting from a diagnostic perspective is a peculiar activity it has, at least in bacteria.
Which is?
Once Cas13 finds and binds to its specific target RNA sequence, it goes into this hyperactive state and starts shredding other RNA molecules nearby, non-specifically.
Collateral damage, kind of.
Exactly. Collateral cleavage. It's like setting off a molecular shredder once it finds its primary target.
And we're actually harnessing that shredder activity for diagnostic tests. That sounds clever.
It is. The Sherlock platform, which came out of Feng Zhang's lab, takes direct advantage of this collateral RNA cleavage.
Sherlock, okay.
You design a guide RNA to target a specific RNA sequence, maybe from a virus you want to detect, like influenza or SARS-CoV-2. You then mix Cas13a, the guide RNA, and a special reporter RNA molecule.
Oh.
Yeah, this reporter is tagged with a fluorescent molecule on one end and a quencher molecule on the other. Normally, the quencher keeps the fluorescence turned off because they're close together on the intact reporter RNA.
Okay, fluorescence is off.
But if Cas13a finds its target viral RNA in the sample, its collateral shredding activity kicks in and also chops up the reporter RNA.
Ah, separating the fluorescent molecule from the quencher.
Precisely. When they get separated, the fluorescence is turned on and starts to glow. So a fluorescent signal tells you that the target RNA was present in the sample.
That's brilliant. Simple signal readout.
Yeah, and Sherlock has been further refined for better sensitivity, and it can even be implemented on simple lateral flow strips, like a home pregnancy test. Like, it's a very accessible diagnostic tool.
And Detector is another CRISPR-based diagnostic platform. Uses a different enzyme.
Yes, Detector, developed by Jennifer Dude in his lab, uses a different CRISPR enzyme called Cas12a. Now, Cas12a is primarily known for targeting DNA.
Right, like Cas9.
Correct. And in the context of Detector, it's engineered to detect specific DNA sequences, again, maybe from pathogens or indicating specific genetic mutations. Similar principle to Sherlock, it employs a reporter molecule.
But a DNA reporter this time.
Exactly. A single-stranded DNA molecule that is also fluorescently quenched. When Cas12a finds its target DNA sequence, it also exhibits a kind of collateral activity, but it non-specifically degrades single-stranded DNA nearby.
So it chews up the DNA reporter.
Right, releasing the fluorescence. Same outcome. Signal indicates the presence of the target DNA. And interestingly, as Adjane mentions regarding SARS-CoV-2 detection, these platforms can be combined. Sherlock AV2, for instance, can use different Cas enzymes in the same reaction for multiplexing.
Multiplexing, detecting multiple things at once.
Yeah, exactly. You could use Cas12a to look for one virus's DNA and Cas13a to look for another virus's RNA, all in the same sample, simultaneously.
So CRISPR isn't just about editing anymore. It's becoming this incredibly powerful and versatile tool for rapid, sensitive disease diagnostics. That's a huge shift.
It really is. Moving from the lab bench towards point-of-care applications.
Okay, our final major topic is truly on the cutting edge. Live cell RNA imaging using CRISPR-Csm complexes. This sounds like we're literally getting a movie of RNA molecules inside cells.
It absolutely is aiming for that. The Nature Biotechnology article you shared really dives into this. It highlights the significant challenge of trying to visualize individual, naturally occurring RNA molecules inside living cells.
What's the main difficulty there?
Well, traditional methods often lack the resolution to see single molecules clearly, or they generate too much background noise, making it hard to pick out the real signal, or they only work in fixed, dead cells.
Right.
But this study introduces a new technique called SEM-Live-FISH. That stands for single molecule live cell fluorescence in situ hybridization.
So live F-MAD issue.
And it cleverly leverages the type 3A CRISPR system from the bacterium Streptococcus thermophilus. The key is using multiple guide RNAs simultaneously targeting the same RNA molecule.
Multiple guides. How does using multiple guides help us see individual RNA molecules more clearly in a living cell?
Okay, so the basic idea, building on older SEM-FISH techniques used in fixed cells, is to essentially decorate the target RNA molecule with lots and lots of fluorescent tags.
Paint the RNA with light.
Kind of. The CM-TOM complex itself is made of multiple protein subunits. As detailed in the Nature Biotechnology paper, it contains multiple copies of a protein called CSM3. In this SEM-Live-FISH system, they engineered these CSM3 subunits to be linked to GFP, that bright green fluorescent protein.
So each CSM complex already has multiple GFPs built in.
Exactly. Now, if you use several different guide RNAs that target different sites along the same messenger RNA molecule, you recruit multiple of these multi-GFP CSM complexes to that single RNA molecule. All those GFPs clustered together dramatically amplify the fluorescent signal originating from that one RNA.
So it makes the single RNA molecule shine much brighter than the background.
Precisely. Bright enough to visualize individual RNA molecules as distinct spots within the complex environment of a living cell.
That makes perfect sense. More tags equal brighter signal. The paper mentions specifically targeting NOTCH2 and MEP1B transcripts. What were the key insights they gained from actually watching these RNAs move around in real time?
What's really groundbreaking here is that they were able to directly observe distinct and dynamic localization mechanisms for these two different messenger RNAs. It wasn't just random diffusion.
What did they see?
For the NOTCH2 mRNA, they saw movement patterns that strongly suggested co-translational translocation at the endoplasmic reticulum.
Meaning
The RNA molecule is being actively transported to the surface of the ER membrane at the same time that a ribosome is moving along it, translating it into protein. The protein is likely being fed into the ER as it's made.
Wow, watching protein production and localization happen together.
Yeah. And in contrast, the MAP1B mRNAs showed very different behavior. They exhibited clear directional transport out towards the periphery of the cell.
So completely different trafficking patterns.
Exactly. These are fundamental discoveries about how different RNA molecules are actively sorted, organized, and moved within the complex, crowded environment of a living cell. It's not just floating around randomly.
The Nature Biotechnology paper also draws a comparison between using this CRISPR-Csm system and using Cas13 for RNA imaging. What are the main advantages of Csm for this specific job?
The paper highlights several key benefits. First, as we just discussed, the chiasm complex's structure, having multiple GFP-linked CSM3 subunits, inherently leads to a brighter signal per target RNA molecule compared to using a single Cas13 protein tagged with GFP.
Brighter is better for imaging.
Definitely. Figures in the paper and a related preprint show that Csm is much more effective at robustly labeling even challenging targets, like the long non-coding RNA Chiaseti, which apparently Cas13 struggled to visualize effectively.
Okay. Any other advantages?
Yes. A second major point is that the Chiasm complex seems to exhibit a significantly higher binding affinity for its target RNA compared to Cas13.
It holds on tighter. Essentially, yes. This stronger, more stable binding likely results in more reliable labeling of the RNA molecules over longer periods, which is obviously crucial if you want to track them in live cell imaging experiments that might last minutes or hours.
Okay, so brighter signal and tighter binding make CRISPR potentially better for live RNA tracking than Cas13. We also have a PubMed article that discusses engineering an all-in-one type 3 CRISPR-Cas system for mammalian cells. What does all-in-one mean here, and why is it helpful?
Right. That PubMed paper, along with its related bioRx-proof preprint, describes creating a system where all the necessary genetic components of the type 3A CRISPR-Cas system are encoded on a single piece of DNA, a single plasmid.
All the parts together.
Exactly. That includes the gene for the Cas6 enzyme, which is needed to process the guide RNAs correctly, plus the genes for all the CSM1 through CSM5 subunits that assemble to form the final effector complex.
And putting it all in one plasmid just makes it easier to get into cells. Significantly easier, yeah. Instead of having to deliver multiple different components, you just deliver this one all-in-one plasmid. It greatly simplifies the delivery and implementation of the CRISPR system in mammalian cells, making it more accessible for researchers.
Makes sense. And this all-in-one CRISPR-Csm system isn't just for imaging, it's also effective for knocking down RNA in mammalian cells, correct?
Yes, absolutely. Figures in the PubMed paper demonstrate that this engineered CRISPR-Csm system can efficiently and robustly knock down the levels of specific target RNAs. And it works on RNAs found both in the nucleus and in the cytoplasm of mammalian cells.
And importantly, how does it compare regarding off-target effects? That's always a concern.
That's a critical point. Figures in both the PubMed paper and the bioRx preprint provide compelling evidence that this CRISPR-Csm system exhibits significantly fewer off-target effects compared to both Cas13 and also compared to traditional RNA interference methods like using hRNAs.
Fewer off-targets, that's a big deal.
It's a major potential advantage, yes. This enhanced specificity is really attractive for both basic research, where you want to be sure you're only affecting your target, and certainly for any potential therapeutic applications down the line.
Okay, finally. The Cell and Bioarcs articles delve into the really nitty-gritty molecular mechanisms, how the CRISPR complex actually recognizes and cleaves its target RNA. What key insights did these structural studies reveal?
These studies provided some really crucial high-resolution structural insights into the inner workings of the CRISPR complex. They confirmed, for example, that the complex cleaves the target RNA at very precise 6 nucleotide intervals along the bound region.
A regular cutting pattern.
Yes. And a particularly interesting finding relates to how it gets activated. It turns out that a slight mismatch, a non-complementarity, between a short sequence tag on the 5' end of the cRNA guide and a corresponding anti-tag sequence on the 3' end of the target RNA. That mismatch is actually required to activate other functions of the complex.
A mismatch activates it. That seems counterintuitive.
It does, but it seems to be a control mechanism. This specific mismatch activates the DNA cleavage activity and also the synthesis of signaling molecules called cyclic oligoannilates, or COAs, by the CSM1 subunit. These COAs can then activate other downstream defense mechanisms in the bacterial cell.
So the mismatch acts like a specific trigger signal.
Exactly. The structural papers show how cognate targets, fully matching, and these non-cognate targets with the 3' mismatch actually bind into slightly different channels within the C-endomosome complex, triggering these different downstream activities.
Fascinating complexity.
It is. These structural details, understanding the architecture and how RNA binds, are fundamental to figuring out how this system achieves such high specificity and minimal off-target effects. And that knowledge helps optimize it for applications like RNA imaging or targeted RNA degradation.
Okay, let's just take a moment here to really appreciate the breadth of what we've covered in this deep dive. It's crystal clear that CRISPR technology has absolutely exploded far beyond its initial role as just a gene editor.
It really has become a whole platform technology.
Yeah, an incredibly versatile platform. It's allowing us to not only precisely manipulate the fundamental building blocks of life, but also to visualize processes and diagnose diseases with really unprecedented accuracy and sensitivity. From developing these advanced Cas9 variants offering finer control and the emergence of anti-CRISPR proteins acting as essential safety brakes.
Crucial for safety, yeah.
To the innovative use of Cas systems and CRISPR-paired recombinases for truly large-scale genomic engineering. The level of control we now seem to have is, well, it's astounding.
The possibilities for genome rewriting are expanding rapidly.
And the ability to perform high-throughput screens with CRISPR libraries is just revolutionizing the pace of biological discovery, figuring out gene function on a massive scale.
Absolutely accelerating discovery.
While repurposing dCas9 for activation, repression, visualization, and purification has given us this unparalleled toolkit for studying gene regulation and the intricate architecture of the genome itself.
Yeah, understanding the genome in its native context.
And perhaps most excitingly, maybe, the development of CRISPR-based diagnostics like Detector and Sherlock. They offer rapid, sensitive ways to detect pathogens, genetic mutations, bringing advanced diagnostics much closer to the point of need.
Huge potential impact there for sure.
And finally, this leap into live cell single-molecule RNA imaging with CRISPR-Csm, showcased by Smuth Livefish. It's providing us with this dynamic, real-time view of RNA behavior inside living cells. That promises to unlock fundamental secrets about cellular processes and ultimately disease mechanisms.
Seeing biology in action at that level is just transformative. What's truly remarkable through all of this is how these seemingly disparate applications, editing, controlling, screening, visualizing, diagnosing, imaging RNA, they're all rooted in that same fundamental mechanism. Guide RNA directing a Cas protein to a specific nucleic acid sequence.
The core principle remains.
Right. Each new development just leverages that core principle in ever more ingenious ways, constantly pushing the boundaries of what we thought was possible in biology and medicine.
Absolutely. It's incredible. Thank you for guiding us through this incredibly rich and clearly rapidly evolving field today. It's truly inspiring to see the power and versatility that CRISPR technology now offers.
It's been a pleasure to unpack all of this with you. There's certainly a lot to keep track of.
It really is. And thank you for joining us on this deep dive into the multifaceted world of CRISPR. We hope this has given you a clearer and maybe more exciting perspective on this groundbreaking technology.
Yeah, hope it sparks some interest.
Until next time on Bio Bits and Bytes, keep exploring.